and welcome back to BioBlinkers. In today's video, we'll talk about the transcription in eukaryotes. This topic is very important from NEET point of view, so stay tuned. As you all know that transcription basically means copy. When the information is transferred from the DNA to RNA, this process is known as transcription. When the information is transferred from RNA to protein, this process is known as translation. Transcription, translation, both take place in separate compartment in case of eukaryote. Transcription take place in nucleus and translation take place in cytoplasm. Different RNA polymerase transcribe different RNA in case of eukaryote. RNA polymerase 1 transcribe rRNA, RNA polymerase 2 transcribe mRNA, RNA polymerase 3 transcribe tRNA. A typical transcriptional unit basically consists of the promoter, structural gene and the terminator. In case of eukaryote, the transcriptional unit is monocistronic. Mono means one and cistronic is the alternative name of the structural gene. That means in eukaryote, every transcriptional unit will have only one structural gene. And every structural gene will has its own promoter and terminator. Now let's begin with the transcription process in eukaryote. The promoter in eukaryote will have some conserved sequences, and these conserved sequences are known as data box. These sequences are recognized by the general transcription factors TF2D, TBP, TF2D, TAF, TF2A, TF2B, TF2F, TF2E and TF2H, these are the name of the general transcription factors. The main function of these general transcription factors is in the recruitment of the RNA polymerase 2 to the promoter. That means the RNA polymerase 2 is indirectly bind to the promoter and the connecting links are the general transcription factors. You all know that DNA is a double-stranded structure. It had two strands, coding strand and the template strand. When the RNA polymerase bind to the promoter, then the RNA polymerase 2 will use the template strand of the DNA to synthesize the mRNA. General transcription factor and RNA polymerase 2 together form the pre-initiation complex. One of the general transcription factor will add the phosphate group to the RNA polymerase 2. Once the phosphate groups are added, now the RNA polymerase 2 will leave the pre-initiation complex and start stalling over the DNA. This process is known as initiation. Now, the RNA polymerase 2 will stall over the DNA and a small stretch of pre-mRNA is synthesized. The pre-mRNA is synthesized in 5' to 3' direction. As the RNA polymerase 2 moves forward, the length of the pre-mRNA keep on increasing as the new nucleotides are added to the pre-mRNA, and this process is known as elongation. Now, when the RNA polymerase 2 encounters the terminator or the termination sequences, then the pre-mRNA is released and this process is known as termination. Now, this pre-mRNA will undergo some post-transcriptional modification. As you know that a DNA consists of the coding sequence and non-coding sequence. Coding sequence are referred as exon and non-coding sequence are referred as introns. And introns does not code for any functional protein. As pre-mRNA is formed from DNA, so it also has exon and introns. And removal of the introns during the processing of pre-mRNA, the process is known as splicing, which is one of the most important post-transcriptional modification in eukaryote. Once splicing is done, now at the 5' end of the pre-mRNA, the 7-methylguanosine triphosphate cap is added, and this process is known as capping. Another modification occurs at the 3' end where the adenylate residues are added or the poly-A tail is added, and this process is known as tailing. So, the post-transcriptional modification will include the splicing, capping and tailing in case of eukaryote, because of which the pre-mRNA is now converted to the mRNA or the mature messenger mRNA. Now, this mRNA, once the modification is done, will move from the nucleus through the nuclear pore into the cytoplasm for the translation. Now, let's discuss the functions of capping and tailing. First function is the protection. Capping and tailing protects the mRNA from the exonucleases which are present in the cytoplasm. Second function, they help in the transport of the mRNA from nucleus to cytoplasm. Third function, they serve as a recognition signal for the translation. Now let's discuss the difference between the prokaryotic transcription and the eukaryotic transcription. First difference, prokaryotic transcription takes place in cytoplasm, whereas the eukaryotic transcription takes place in nucleus. In prokaryotic transcription, single RNA polymerase is transcribing all the 3 RNA, whereas in eukaryote, different RNA polymerases are transcribing different RNA. In prokaryote, general transcription factors are absent. That means the RNA polymerase II are directly binding to the promoter. There is no connecting links are present, whereas in eukaryote, general transcription factors are present, because of which the RNA polymerase II is indirectly binding to the promoter, and the connecting links are the general transcription factors. In prokaryotic transcription, post-transcription modification is absent, whereas in eukaryotic transcription, post-transcription modification are present. That means you will observe the splicing, capping and tailing in case of eukaryotic transcription. Thank you for watching this video. If you like this video, please don't forget to subscribe to my channel